To this webinar. Welcome from me. My name is Jane Knight and I'm an MSSO instructor. My official title is a clinical associate, and I'm also joined by my colleague Leander Mould, who's been speaking with you already. And as Leander said, we will stop throughout the session to ask questions. Welcome back if you were with us last week for our other webinar, which was data analysis and query building. So if you're not familiar with the general data analysis, that recording is on the YouTube channel, as are other recordings of that session by colleagues. So if you feel you need a refresher of the background and you weren't with us last week, feel free to go and listen to those. But if you are back with us today, we're going to be doing standardized MedDRA queries, or SMQs, and focus on that. We have 2 hours together to talk about SMQs, look at them in the browser and ask questions. So do take this opportunity to make your comments, raise your questions, and just enjoy the time seeing what SMQs can do. The first slide is our standard slide explaining that MedDRA came about in the mid-1990s. It was an ICH initiative, and at the time, the MSSO, the Maintenance and Support Services Organization, that's the organization that we both work with, that organization was put in place on contract to maintain, develop, and distribute MedDRA. The activities of the MSSO are overseen by a steering committee, and that has representatives from across the whole of the ICH regions and both the industry and the regulatory authorities. You see some of the members listed on this slide, and there are others who are observers and are just recently joining the steering committee. And you see the details on the slide there. The second one is our standard copyright notice explaining that MedDRA is owned by ICH, as is the material we're going to use today. So if you do use the material or quote it in any way, you must acknowledge ICH as the owners. If you change it, you can't assume that you have permission or that the change is approved by ICH. And if there are any third-party slides, I'm not sure there are today, but if there are any that we've received from other people, you can't quote them without their permission. So that's everything on this slide. So now let's look at what we're going to cover today. We're going to review the content of the MedDRA data retrieval and presentation points to consider document. Now, if you were with us, you'll remember that we spoke quite a lot about this document last week, thinking about analysis in general. So I'm going to cover this quite briefly because we did cover it in more detail, but the SMQs themselves do also come into this document. So it's a reminder to you that if you want to see a refresher of some of the information I'll give you today, this is one of the important resources that you can go back to learn more about how to use the SMQs and the implications of doing so. There's a list there of what we're going to cover in relation to the SMQs, and there's another slide here which goes into more detail. We're going to look at a demonstration of the SMQs, applying them to some data, and I'll show you how you can do that within the MedDRA browser. We'll look at, in theory, how SMQs can be applied both to clinical trials and pharmacovigilance, and I'll give you 2 fictional scenarios showing how SMQs can have been beneficial to an organization, and we'll wrap up towards the end with a creation of customized searches. That sounds like an awful lot in 2 hours, but I am well confident that we will have time this week. I know we had a comment last week, there wasn't enough time to answer questions, so we really are going to do our best to get through your questions and give you the information that you need today. Okay. So with that, I'm going to switch off my camera because it can be distracting for you to watch me making faces and reaching for my water at various times. So I'm going to switch it off. There we are. So this slide explains the polling application we're going to use. It's called Poll Everywhere. And you can access the polling and vote in a couple of questions, not too many, but if you scan the QR code with your mobile phone, that's the easiest way. It will connect you directly to my presentation. Alternatively, you can open a browser on your internet. So it would be a browser like Safari or Edge or one of those. And in the URL, you type in polev.com. When you see this slide, the picture, the screen on the second, the right-hand screen, you enter my name, Jane Knight 855. That's the username. It's completely anonymous. You don't put your own name in, but that will allow you to link in to my presentation and join the polling. So by the time I've explained that, I'm sure most of you have done this. The QR code will appear again on the various slides where we have a poll. And if it seems like your phone gets stuck, press the refresh button and it may well refresh without needing to rejoin. Happy with that? Good. So here's a poll for you. Where are you joining from today? Let's make sure this is working. Oh, yes, I can see some of you are across Europe. Oh, lots of you across Europe. Someone from Brazil. I think you were with us last week, if I remember correctly. India again, maybe. If it wasn't you, it was someone else from Brazil. Is that New Zealand? Or is that Australia? Wow, across the whole way across Europe. I can't even make the countries out there. I can guess, and I'm sure I won't be wrong. Spain. Again, someone else from Latin America. Oh, someone write down. Okay, so well done. Wherever you are in the world, you are so welcome to this. I always feel this is a privilege. I say that often, but to see this map and to think about how technology allows us to do this and meet up just as if you're in the room with me, I feel it's such a great privilege. So welcome, welcome. So now the data retrieval and presentation points to consider document. So in general about this document, it provides data retrieval and presentation options for the industry or regulatory purposes. And you'll remember last week we went through some of these options, not the SMQs. We talked last week about hierarchy of MedDRA, multi-axial links, and how these can be used as an option for data retrieval or presentation. This document is most effective when it's used in conjunction with MedDRA term selection points to consider. Well, as we've said before, if you don't code your data systematically, consistently, and accurately, you're going to have mixed results when you try to retrieve or see patterns in your data. So if you haven't been consistent, then you're not going to see the trends and the patterns that lie within your data. And this document is recommended to be the basis for your own individual organization's data retrieval conventions. It comes out once a year, and you can see there the front cover of the document. I've added this year's date, last year's date, because that was the most recent release of the document to show you how it would look. In March this year, we'll have the next one, which would be release 3.26. So it is developed by a working group of the ICH Management Committee. So this is the more senior committee within ICH overseeing MedDRA. It's updated annually in March. There are translations of it in a number of languages, as you see on the screen. There are also condensed versions for some of the other MedDRA languages. And you see by the asterisk, those languages with a summary or condensed version are on the screen. That we have MedDRA now in 27 different languages, different translations. In recent years, we've produced a number of translations quite quickly, and these are in support of an electronic prescribing initiative by the EMA. So because we've produced the translations to meet the needs of this initiative, that's the main purpose of the languages. Our focus has been getting them out there and available for people. So that's why there aren't the documents associated with all the MedDRA languages, in case you wonder, in case you want to use one of the other languages, that's why. And just getting back to the document, it is available on both the MedDRA and the JMO, the Japanese Maintenance Organization websites. You'll remember that MedDRA is maintained by the MSSO for all languages apart from Japanese, which is maintained by our partner organization, the Japanese Maintenance Organization. So as this is what we did cover last week. So if this is new to you, then I would point you back to the session from last week. The beginning of the document talks about some general principles relating to data in general. Firstly, the quality of the source data. So if your data are of poor quality being received, and when you code them, you don't really understand what's being reported, you're not going to have good quality coding. And ultimately, you're going to not have good quality analysis. So it's going to be difficult to see a pattern and retrieve the cases that you may be looking for when you use the SMQs. The next principle is the importance of documenting your data retrieval and presentation practices. I sometimes say to my classes, imagine you're being inspected by an authority, a regulatory authority, and they come along and say, well, how did you do this? What was your rationale? How did you code? And so this is something to be aware of. Also, you might move on to another role. You might forget what you did when you come back to an analysis in a year's time, for example. So document as much as possible. Do not alter MedDRA. MedDRA is a standardized terminology with a predefined term hierarchy. So you mustn't make any ad hoc structural alterations, that would include changing the primary SOC when you code data. Be aware of your organization's specific data characteristics and know how you store the data, how you use the levels within your database. Also know your IT system, know what is possible within your organization. Be aware of the characteristics of MedDRA. And again, I point you to the hierarchy, the multi-axial links, and then also MedDRA versioning. We know that MedDRA comes out twice a year, so know what version of MedDRA you used for coding and match that version to the version you use for the SMQs or for your analysis. That's all in the beginning of the document. Then it goes on to discussing general queries, and this is what we did last week. Standardized MedDRA queries, our topic for today, and customized searches, which I'll also address with you today. So MedDRA versioning, it is updated twice a year on the 1st of March, where any of the 5 levels could be updated. That's called a 0.0 release. So we're now waiting for version 29.0. On the 15th of March, we'll have the translations. On the 1st of March, we'll have the English version. And similarly, in September, on the 1st of September, we'll have the English 29.1. The 15th of September, we'll have the translations. And in September, we only see changes to the LLT and PT levels. The version used in your data retrieval and presentation should be documented. There are some resources there listed on the screen for you where you can find out more information about versioning. And I'll show you when I go to the website where you can find these documents. What's new? We do a webinar with each new release of MedDRA, as well as documents explaining what's new in the new version of MedDRA. There's a best practice document that covers lots about versioning, especially in relation to clinical trials, if that's an interest for you, as well as pharmacovigilance and general dictionary updates. There's the version report, which is an Excel spreadsheet listing all the changes between the last version of MedDRA and the new one. Later, I'm going to show you where to find that. So make a note if that's something of interest. And there is a MedDRA version analysis tool, which I will also come to in more detail shortly. And as I've already mentioned, what is important is that the terms used for your queries, whether that's a query you built or yourself, or whether it's a standardized MedDRA query, your terms should be in the same version as the data being queried were coded. So that's very important there. If you've got these slides, put a highlighting against that point. Very important to remember. So the MVAT tool. The MVAT tool is available in 2 places via our website. There's a standalone version with the URL on the screen there. It's free for you to use. It's also available within the new web-based browser. So if you, and I'm going to show you this, it's free again in both places, but you can use it. It works exactly the same. It's just that when we released our new web-based browser, we incorporated the MVAT tool as part of the data analysis function within the browser. So that's why it's there. The tool has 3 features. It firstly allows you to generate the version report. This can compare any 2 versions of MedDRA, not just the previous and the recent release. You can go back and look at the difference between, for example, 2 years or 3 years if you're working in clinical trials and you're looking to update your reported data. You can also import your own data and just see the impact of the MedDRA update on your own data. So that's where you can focus onto a subset of MedDRA terms that you used for coding. And thirdly, you can look at the history of individual terms. That's something that you do in the standalone version, and you do it in the MedDRA browser under a history function, but it works exactly the same. So that's the MVAT tool. It's available in any of the MedDRA languages, both the interface and the report of the output. You can run to see changes, including the supplemental changes to MedDRA. Now, if you're not familiar with this word, supplemental changes, it refers to new terms which have been approved by the MSSO physicians to be released in the new upcoming version. So at the moment, there are new terms that have been added to MedDRA, but they haven't been released yet. They're waiting for the 1st of March to be released in the new version 29.0. So this is what we call supplemental terms. They've been added, they've been reviewed and approved. They're just waiting for release. When you run your report using the MVAT tool, you can run it on secondary SOC changes to see the secondary SOC changes as well. This is the multi-axial links within MedDRA, and we don't have time to go through all the functions of the tool today. So I have listed there on the screen that there is a 2-part webinar. If you're interested in using MVAT, you can have a look at this, two short webinars that together show you how the tool works and how you can use it. There's a link there to our training materials page. So I'm going to pause there. It's a good point to pause. Leander, do we have any questions so far?
Thank you, Jane. Currently, there's no questions, but thanks very much for checking in.
Thank you. So let's move on now to think about standardized MedDRA queries, or SMQs, our main topic for today. What is a query, first of all? And you'll be familiar with this slide from last week if you were with us. So on the left-hand side, we see that we're working either in clinical trials, we have a clinical trial database, or we have a safety database. In either situation, we have cases stored in our database, patient reports. Those reports are coded to LLTs in MedDRA, and that's what we see on the clipboard with a case there. And so we come along with our query. Our query is a list of preferred terms. Queries are built at the preferred term level. So it's a list of preferred terms that we think represent that condition of interest. So if you think a patient might have a particular condition that we're interested in, they could present and be diagnosed with that condition, and then they would have an event coded to the diagnosis of the condition, or they may just appear with signs and symptoms or abnormal lab results which are indicative of that condition or a procedure. And so we could have PTs in our list which relate to signs and symptoms or procedures or lab results or any of the MedDRA terms. Wherever they come from, those preferred terms break down into LLTs, And when we run our query against the database, what we're doing is we're coming with our list of preferred terms that we're interested in, and we're searching for patients who have events coded to the LLTs that sit below our preferred terms of interest. So that list of preferred terms could be a query that I've built, and this is what we did last week, or it could be a standardized query, a standardized list of preferred terms that together, those preferred terms all relate to the condition of interest. So it could be an SMQ. If it's not clear, then let me know and I'll come back and go through that again. So then in text form, what is an SMQ? Following on from what I've just explained, an SMQ is a group of terms from one or more MedDRA system organ classes related to the medical condition or area of interest. And these preferred terms could be signs and symptoms, diagnoses, syndromes, physical findings, lab tests. And the whole purpose is to help us to identify patients who have the condition we're interested in or may have that condition, and the case needs further review. So it's intended to aid us in case identification, and it does it in a standardized way, because if we're looking for a particular condition of interest and there's another organization doing a similar search for that same condition among their patients, they're using the same standardized list of preferred terms. And this is great. This means we're consistent, it's efficient for us. We don't have to build the query, we're using an SMQ. And from a regulatory authority perspective, they can easily compare the results they get sent from one organization with another. And when you tell them you've used an SMQ, they know instantly what preferred terms you've looked for when you ran your search. So it's all about standardization, consistency, transparency, and efficiency, really, all for patient safety. There are 110 level 1 SMQs in production as of version 28. So I've just mentioned we're currently using MedDRA version 28.1, but our training materials are updated once a year in March. That's why this slide relates to version 28, but there are still 110 SMQs today. Level 1 relates to something I'm going to explain in a few slides, so stay with me and I'll explain that shortly. You see here just a few of the examples, and you can see immediately that the SMQs were built for drug development. So typical red flags in drug development: hepatic disorders, hypersensitivity, an anaphylactic reaction, as well as some SMQs that have been built because they may be a common adverse reaction for particular types of drugs, maybe osteonecrosis, peripheral neuropathy, pseudomembranous colitis, for example. Now, here's a poll for you. What PTs could be included in an SMQ? What could be included in this, in the SMQ? What types of preferred terms? You can click on the pictures. If you don't want to join, ask yourself the question, have a look, and you can test yourself and see how you go. So if we got everything clicked, we see
Jane,
some
can
of
I
the
interrupt you just for
yes,
a moment?
of course.
Oh, thank you. We did have a query. So the cases that are saved inside MedDRA are only from clinical trials. So I know you were talking earlier on your previous slides about clinical trials, but could you just clarify that when you're...
So I think your wording of your question, their cases saved, so cases don't get saved within MedDRA, they get coded to MedDRA. So I think that's probably what you meant. So just for everyone online, just to get the wording correct, the cases come in, the patient events are coded to MedDRA and they're stored in your database. So that could be a clinical trial report, it could be a spontaneous post-marketing report. SMQs can be used both for clinical trials and pharmacovigilance, and I have a slide later giving you a few examples of how they can be used, both clinical trials or pharmacovigilance. So don't switch off and run away if you're working in pharmacovigilance. What we're going to cover today is equally applicable to both areas of work. I hope that answered your question. If not, come back and clarify and we'll try again. So I can see now, look, you've obscured the writing. I can't even pick them out. Now, I think you can see for yourself. What we're saying is preferred terms could be from any area of MedDRA. They could be reports of death, they could be toxicology, they could be medical history, syndromes, they could be lab results. As I say, I can't even read them underneath. So well done. Thank you for participating and just illustrating. So we all have clear in our head the broad scope of MedDRA and how I think this also illustrates for me how the benefit of an SMQ from an efficiency perspective. Other experts have been through MedDRA, the whole scope of MedDRA. They've picked out all the relevant PTs for me, so I don't have to do that when I'm thinking about a condition of interest. It's already been done in a standardized way. I can take it off the shelf. So thank you for that. Now, here is a valuable resource, and this is where I think I'll pause and I'll go to the website to show you where some of these documents are. This is an introductory guide to standardized MedDRA queries. It's available with each new release of MedDRA, which is why you see the XX. I haven't specified what release it relates to. And when you download the new release of MedDRA, the introductory guide is with the files, and they're also stored on the website that I'll show you now. So this document is important. This document goes through information relating to every SMQ one by one. It shows the structure of the SMQs, how it was built, the sources that were used, the medical references that our teams used in building the SMQs, and it can help you in understanding the output and how to use them. So we can't go through 110 of them today, so I would point you to that. So that's a good point for us to pause, and I'm going to the website. So if you've got any questions relating to finding things on the website, now's your chance to send them in to Leander, and she can direct me to where you want me to go. So this is the MedDRA homepage, www.meddra.org. I'm going to go how to use. That along the top you see the tab support documentation. Now support documentation is in two sections. The blue banner is where we store the points to consider documents and the best practices document. So these are documents that don't get updated with each release of MedDRA. We know the points to consider comes out once a year. Best practices is updated as and when necessary, as is the companion document for coding. So this is where we see the MedDRA term selection points to consider. That's our coding guidelines document. And we also see the data retrieval and presentation points to consider. I mentioned that. That's the earlier today. That's where we see the data options. It explains how the features of MedDRA, like the hierarchy, the multi-axial links, how to build a query, or how to use the SMQs. And it's available in three formats: PDF, Word, and an online navigable version. That's so that if you want, you can incorporate it into your own SOP. Then we have a red line version showing us the differences with each new release of MedDRA. To be honest, not much gets updated in this data retrieval and presentation. There's updates if there are new SMQs released. And then we see here underneath the listing, and there's the best, before I move on, sorry, there's also the best practices document. The best practices document, this is where I said it's a good document for clinical trials. It also has versioning information relating to pharmacovigilance. Let's have a look at the index so you can see. It talks about MedDRA, and this is best practices in using MedDRA in general. This is where if you ever get challenged on maybe you're working with a clinical team or a medical reviewer and they want you to change coding to a different SOC, this is a good point. This is a good place to as a reference, as a justification for why MedDRA places the terms and why the SOC shouldn't be changed when you code, but instead to use multi-axial links. Anyway, I digress. Here we see the version control, semi-annual version control explaining versioning for clinical trials. So I pointed out this is particularly useful for clinical trials, but then we also have the methodology, which applies equally to both clinical trials and pharmacovigilance, and also some information about the supplemental terms. Remember, I explained what these were. They're the terms that have been approved and they're waiting to be released. So that's that document. Let me go back to the website. So now we have the list of versions. So if I click on MedDRA version 28.1 from last September, you'll see this is where you can find the introductory guide to MedDRA and to the standardized MedDRA queries, intro guide, and the what's new document that I mentioned. So let's open that one and have a quick look at the index. You'll see what I mean. So here are all the individual SMQs listed. The introduction talks about SMQs in general, and they're hyperlinked, so you can access one of them if we want to see, for example, one of our most complicated ones. We could say hepatic disorders, or we could use one of a more simple one. Let's pick a simple one for now. Let's go to arthritis. Should we look at arthritis? So it tells us the definition of arthritis that's been used in building the SMQ, lists some of the terms, the typical symptoms, types of arthritis. So inclusion and exclusion criteria would be described here. There they are, the inclusion, exclusion. There's the reference that was used. It tells us there a bit about the structure. That's a simple, just a basic list of PTs. And you'll see when I talk more about the characteristics, what I mean by some of the SMQs being more simple and some being more complex. So that's the document, and this is where you find it. Have we had any questions, Leander, relating to the website or how to find specific resources?
Thank you, Jane. We did have one that was about the introductory guide, and then you went to it. So thanks very much for that.
Great. So let's go back to our presentation for today. So now I'm going to talk about the SMQ data characteristics. This is what I was saying about some being more complex. You'll see what I mean. If you want to follow along, we've brought along here a demo password for this week. And this is the password for it. Leander's put it in the chat, so take it from the chat. And I'm going to use the new web-based browser in my demonstration of the SMQs. So MedDRA term inclusion. So this is what we've already said today. SMQs are constructed at the PT level. That's because the preferred terms are a single unique medical concept. There's no duplication at the PT level within MedDRA. And beneath the PTs are LLTs. And we can see by the numbers of terms at each level here, how at the LLT level, there are a number of synonyms or other ways of expressing the same concept. The preferred terms are our medical concepts, and at the LLT level, we have synonyms. So that's why we want to be as efficient and clear in building our query. So we build it by selecting preferred terms. And that's what the SMQs are, groups of preferred terms. The LLTs underneath are automatically included. Now, this, I think, is the most useful and the most important slide in today's presentation. So please do question me if it's not clear. The preferred terms within the SMQs are designated as either narrow in scope or broad in scope. Narrow preferred terms give us specificity. So these are the preferred terms which are highly likely to be used for coding the condition of interest. So if we're thinking about, let's go back to our example of arthritis. If a patient has a report coded to arthritis, they are highly likely to have arthritis. That goes without saying. So it may be a very, very strong characteristic symptom, it might be a type of arthritis, but it would be a preferred term that is highly likely to be the condition of interest. That's known as a narrow scope preferred term. It gives us specificity. Broad scope preferred terms give us sensitivity. These are maybe symptoms or lab results which could be indicative of the condition, but not necessarily so. It could equally be indicative of a different condition. So if you think of some of the symptoms that may be the condition of interest, but maybe not. And this is sensitivity. What I mean here is making sure I don't miss any cases when I run my search using the SMQ. So if I have the option then, when I run the SMQ, I can look just for those patients coded to events under the preferred terms that are narrow. So highly specific search, narrow in my focus, or I could choose to run a broad scope search. And remember, the broad scope, I want to look for the broad PTs and the narrow PTs together. That's what you see. There's no point just looking for symptoms or lab abnormalities without looking for patients with a diagnosis of the condition. So you either can run the whole SMQ with a broad search, or you can run a narrow focus search. So we sometimes get the question at this point saying, well, how do I decide? How would I know? It's your choice. You know your data. You know where at the stage you are in your analysis. Is it late on in the analysis where you know the drug's been around for a long time, you know the safety profile, and then you can be more focused it's more specific in your search. But otherwise, if it's earlier on and you're not really clear on the safety profile of the drug, in clinical trials, you might be screening, then you want to be as broad and inclusive as possible because you're looking for patterns. So that's where you make the call, and it is purely optional whether you use the specific search, narrow only, or whether you run the whole SMQ with all the PTs together. So let's have a look at that. This is an example, lactic acidosis. This is the whole SMQ. It's a small SMQ because it fits on my slide. And you can see there are 3 narrow terms. Blood lactic acid increase, so a lab abnormality, very highly connected with lactic acidosis. Hyperlactacidemia, high level of lactic acid in the blood, or the name of the condition, lactic acid itself. So that lactic acidosis. So that's the narrow terms, highly likely to bring back relevant patients. Whereas if we include the broad search and we look at the broad search terms, we see abnormalities in lab results or symptoms there. A patient with abnormal blood gases may have lactic acidosis. They may have chronic obstructive airways disease, or they may have something else. A patient with acidosis may have lactic acidosis, or it might be a different type of acidosis. So this is why these are broad. They may be the condition, but maybe not. Sensitivity. And it's all a balance between the sensitivity and specificity. So now I'm going to go to the browser for a change of scene. So let me show you this concept. I'm going to go through one concept at a time, one characteristic, and then I'm going to demonstrate it. So this is our website. So let me show you how I'm going to access the browser. How to use tools. On the tools page, there is another page relating just to browsers, but I'm going to go to our new web-based browser. So you're probably aware, we have 2 types of browser. We have a desktop browser, which you download onto your desktop, and we have a web-based browser that we store on our server, and you just access it via a URL. I'm going to use the web-based browser. The benefit of the web-based browser is that we update every 6 months. We have all the languages, all the versions on our server. You don't have to manually import the new release of MedDRA. But the desktop advantage is you don't need an internet connection. So it's handy to have as a backup, or if you commonly work where you don't have an internet connection. So those are the two types. We also have been updating our browsers recently. So we have an older legacy browser, which has a different appearance from the new, more streamlined browser. And that both the desktop and the web-based are available in a legacy version and an updated new, more streamlined appearance version. So I'm going to use the more streamlined, cleaner screen, new web-based browser. So now you see at the top, I'm going to switch now from the SOC view. You see at the very top on my bar, I've got SOC and SMQ in two gray buttons. I'm going to switch to SMQ. If you do open the browser, you might notice that yours looks slightly different. I've got an MSSO account, so I'm going to switch back to version 28 because I've got the 29 already in preparation for the release. So that's why mine automatically went there. And you can use any version of MedDRA. You can access any version using the drop down. So let's go to 28 to match the slide material. You can open different languages, and along the top, I've got these icons. This is because I've customized using the settings, the gear, I've customized my screen. And this, what you're seeing here, is the equivalent of the desktop, of the old legacy web-based browser or desktop browser, with both the filter for the SOCs on the right-hand side and the icons along the top. So I've got, that's my choice. I've customized it. I've got everything available at a single click, and you can do this if you wish, including the functions that you use a lot. But when you first go into the desktop, when you see the main screen, I should say, you see it's very clean and clear. That's because we allow you to customize to suit your own requirements. Okay, so let's do this. SMQs. So SMQs, let's go back to where we were. We were talking about broad and narrow and how they appear in the browser. So we can see I've opened acute renal failure. Acute renal failure has a list of blue PTs. The blue PTs are the narrow PTs, highly likely to be the condition of interest. So a patient with a diagnosis of renal failure is highly likely to have acute renal failure. Patient on hemofiltration, hemodialysis is highly likely to have renal failure. But if we scroll down, some of these lab results, for example, a patient or procedures, patient who's had a renal transplant may have had renal failure, maybe not. It may have been for another reason. A patient who has protein in their urine may have had renal failure, they may not. Albuminuria, they may have had an infection or some other reason, proteinuria, for example. So that's the broad and narrow. If you open the PT, the LLTs are underneath it, but we don't need to look at them today. If you forget the legends, what the blue and red mean, I'm now going to the three lines in the top left-hand corner. So this is what we call the hamburger, because it looks a bit like a sandwich. And that one of the options is legend. You see top left, and here we see the colors for the PTs. That's when we're looking at the SOC, and then underneath we see the SMQ legend. So we can see that blue means narrow scope terms, and I'm going to talk about active and inactive in shortly. What this is that when the preferred terms are added into an SMQ and the SMQ is put out into production, we might receive feedback from our users that says, actually, when we use these SMQs, some of the PTs are not helpful. So rather than take them out, what we do is we just make them inactive. So if we agree with you and you've done all the testing for us, so you're giving us this feedback, and maybe that we agree with you, and we can then make the preferred term inactive. It's still current in MedDRA, but it's no longer used within the SMQ, and that's what that means in the legend. So you can see there. Okay, so that's broad and narrow. Let's go back. Any questions, Leander, on broad and narrow?
We did have a question that said, why would I select a broad and a narrow? And I said that you'd come on and explain this
I've done
as we
that
get to
already.
it.
I have done that. When would you select broad and narrow? It tends to be, you know, your data. How big is your data set? At what stage? Are you early on at the screening stage when you don't really know the safety profile? Then I want to be more inclusive. But if it's the drug's been on the market for a number of years, I know the safety profile, then you would consider probably running a more narrow search to run the narrow PTs only, because you know the safety profile. That tends to be the difference. But remember, while you vote on this one, I just remember, when you retrieve the cases, you need to review the cases. You can't automatically assume, just because they've been retrieved, that they are the condition of interest with the broad PTs, because some of them may be symptoms and the patient may have the symptom, but you need to review their case, look at other information to see whether you think they, in your medical judgment, they do have the condition or not, before you then submit the cases as results to an authority. So the SMQ gives you a way of retrieving potential cases, and in some where it's narrow, the likelihood is the patient will have the condition. But if you throw your net very wide, do a broad search, expect to have more work for you in reviewing the cases, and that's natural. That's what's expected, and that's what you can plan for. So what type of PTs will be used for running a broad search within the SMQ? Is it only the broad PTs? Is it broad and narrow PTs? Broad and narrow plus inactive, broad scope plus a narrow selection of PTs? I've got a few options there. Thankfully, the majority of you did get this one right. When I run a broad search, I want to include both the broad and narrow PTs. I need to retrieve patients who have a diagnosis of the condition or an event which is heavily associated with the condition, as well as patients who have symptoms or lab abnormalities or procedures which may be indicative of that condition, but maybe not. So running a broad search is where I need to include both broad and narrow scope terms. I don't want the inactive PTs. Those are PTs which today, in the latest release of the SMQ, feedback has told us that they're not beneficial, those PTs. So we've switched them off, we've made them inactive. And then the last point here, the broad scope plus a selection of narrow, this is customizing of the SMQ. And this is something we're going to look at at the end of our session today, when we look at customization of the SMQs. Okay, well done, well done. The next feature is algorithm. Now, this is what I was saying, some of the SMQs become more complex. Only some SMQs utilize algorithms. Out of the 110, 10 of them have algorithms. And the idea is it gives better case identification among the broad search terms if we select the cases by a defined combination of selected terms. So what this is saying is, if I look for patients who've got two symptoms and both symptoms are related to the condition, I'm more likely to have a correct retrieval of relevant patients by combining symptoms. It's like when you search on a browser, an internet browser, for example. If you're in Google and you're looking for something, the more words you put in, the more refined your results will be. I often give the example that a patient goes to a doctor and they say, I've got a headache. At that stage, the doctor's thinking, well, it could be lots of things. It's only as the patient elaborates and gives more symptoms, I've got a fever, I've got a rash, I've got photophobia, I've got a stiff neck, the doctor might then think, well, this might be meningitis. So it's the same with making a differential diagnosis. The more symptoms the patient has, the more accurate the doctor will be with their diagnosis. It's exactly the same with retrieving cases. If we look for patients with a combination of relevant symptoms, and that's a defined standard combination of symptoms, if we look for this, we're more likely to have accurate retrieval, and that's less work for us in reviewing the cases. Let's look at an example and it will show you what I mean. Here is anaphylactic reaction. So we have the narrow terms. I'm calling these category A. So you see them there on this in the graphic. Anaphylactic reaction, anaphylactic shock, anaphylactoid shock. Kunis syndrome is a kind of allergic angina. So things which are types really of anaphylactic reaction. And then on the right-hand side, you see we have the symptoms, and these are grouped into categories. Category B are the upper airway and respiratory symptoms like asthma or bronchial edema. Category C are the skin, angioedema, urticaria, and other skin type symptoms, allergic edema, erythema. And category D are the cardiovascular or hypertensive symptoms. Now, I'm not seeing all the PTs for the categories, just a few of them here, but you see the idea. So what the algorithm says is when I run the SMQ, retrieve any patient who has an event coded to the terms in category A, any one of those, or retrieve any patient who has two symptoms or lab abnormalities or whatever it may be, other events, coded to two categories. Either someone with a respiratory problem like asthma, together with erythema or angioedema, they would then qualify for retrieval when I run the SMQ using the algorithm. I would retrieve a patient who may have allergic edema and low blood pressure, because by having two symptoms by different body systems, they're more likely to be related to the condition of interest, anaphylaxis. So it's optional. The category terms are just broad PTs. If you want to just run the SMQ as broad and narrow, you can. You don't have to use the algorithm by combining symptoms, but it's there for you to help you in focusing your search. This is the logic. If your brain works by better by seeing it as a flow chart, we could say, well, firstly, does the patient have an event coded to A? That's the narrow preferred terms. If so, we review them. If not, we ask the follow-up question, do they have a symptom from list B? If we do they have either one from C or one from D? If not, and if they only have one symptom from B, we wouldn't review them. We wouldn't retrieve them, we wouldn't review them. And then we would go on and say, do they have something from category C or category D in combination? And that's the logic here graphically. Here's a question for you again. So about the algorithm, how would you describe it? Is it intended to increase the sensitivity of a search by using an algorithm? Does it increase the specificity of the search? Does the algorithm apply to the narrow scope PTs? And can the algorithm be used without programming effort? That's an interesting thing we haven't talked about yet. So test yourself on this and see what your thoughts are. If you've got questions at this point, I'll take them, Leander, whilst our class is answering the question.
Thanks, Jane. We had one that said, can you export the SMQs?
Yeah, I'll show you when we're in the browser that. Yep, you can.
Thank you. And then we also had, at the beginning, you said that different companies can find the same cases because they apply the same queries, but we can always customize. And I know you're going to come on to customize searches. So that's not always true. Is that correct?
No, so every company's got their own database with their own patients in it. But they're looking for a condition of interest. If they're looking for anaphylactic reaction and they're producing an antibiotic, or another company's looking for anaphylactic reaction to their drug and they've got an antihypertensive drug, both companies are looking for the same symptoms. So they're going to retrieve their own patients who have symptoms or diagnoses which are considered to be anaphylaxis. So when they report into an authority, they're saying, well, they're both using the same strategy for retrieving patients with anaphylaxis. It's like a definition, if you like, of the condition, but that definition then breaks down and translates into MedDRA preferred terms. And so if you imagine, rather than the companies producing different types of drugs, imagine they're both producing an antihypertensive drug. Two companies working on drugs with the same class, they can submit their data and the authority can then compare the relative safety of the drugs based looking for the same condition, using, if they've both used the SMQ, because they've used the same strategy, the same approach to searching for cases with that condition. So it makes comparison easy. It makes co-development of a drug easy between two companies working together on co-developing a drug. So let's see where we are. Hopefully I've answered your question there, but thank you for your thoughts. What have we got? Does it increase the sensitivity? Well, sensitivity is how broad. So it doesn't increase the sensitivity because the broad PTs are in the SMQ. And if I apply the algorithm or if I use them broad, it's the same sensitivity. The PTs are there. So I would say it doesn't increase the sensitivity. The intention, it increases the specificity. It makes the search more specific by only retrieving patients who have two or relevant symptoms or generally or lab abnormalities. So I see there some of you are changing your mind now on that, which is good. So the second one I'd say is the most is the one that I would really say is what I'm trying to get at with this poll. Using the broad terms in general gives us the sensitivity, but by adding the algorithm to those broad terms, it gives us the specificity. Is it applied to the narrow scope PTs? Well, the narrow scope PTs are included in the SMQ when we run it with an algorithm, but that's not the main intention. Mainly they apply to the broad PTs by combining them. And can it be used without effort? So this is something we haven't specifically talked about, but not all systems may have the SMQs readily available. You might have a commercial system that you has the SMQs built into it, a commercial database. It may be you've got an in-house database that's been built to incorporate and to apply the SMQs. That's great. Or it may be in smaller companies or in academia, people may not be able to use the SMQs without programming effort. So not all databases can have the SMQs built into them. And this is why we have the option in our browser, I'm going to show you shortly, to apply the SMQ to your data within the browser. So the last one I would say is also not true. Well done. Hierarchy now, our third characteristic. So look at the picture at the top right, and that will immediately show you what we're talking about here. We know that MedDRA has a hierarchy of the terms. This is not related to that, the 5 levels of MedDRA. This is purely relating to SMQs. Some SMQs develop as a set of queries related to one another in a hierarchical relationship. It's not related to the MedDRA hierarchy. And what happens is that one or more subordinate SMQs, so sub-SMQs, combine to create a more superordinate, that's a big word, a big inclusive, more inclusive SMQ. And it's just like the diagram. You imagine we put preferred terms in each of those boxes and we give them labels. We group the preferred terms for some clinical relevance, and this is what we have. And let me show you to bring it into perspective. Hematopoietic cytopenias could be erythropenia, where red cells are not being produced, leukopenia, white cells, thrombocytopenia, which affects the platelets, or it could be a type of cytopenia where there's more than one of these blood cell types affected. So what we've done is we've put the MedDRA preferred terms relating to erythropenia in that box. This is the sub SMQ, and this is the hematopoietic erythropenia is an SMQ in its own right, but it would be called a level 2 SMQ. Hematopoietic leukopenia is a separate SMQ. It's a level 2 SMQ. That's because it sits below the top box, the all-inclusive hematopoietic cytopenia. That is the SMQ that we would call a level 1 SMQ. Now, what I want to point out, and this is important to understand, is the top level doesn't have any PTs in it itself, except for the ones that sit in the boxes below it. So it subdivides into 4 sections, and when we combine those 4 sections, that is what makes up the level 1 SMQ of hematopoietic cytopenia. So let's go to the browser, because I didn't show you the algorithm that I meant to, and I can show you the algorithm and I can show you the hierarchy together. So let's do hierarchy first of all, because I've just talked about it. So let's open that one, hematopoietic thrombocytopenia, cytopenia. There it is, hematopoietic cytopenia. When I open the SMQ by clicking on it, let's make it a little bit bigger for you. There we are. You can see underneath it the sub SMQs, 4 sub SMQs, and you see there are no preferred terms here hanging at level 1. They're all at level 2. And when I put my cursor on the SMQ underneath it, that's when I open it and I see the narrow blue PTs, the broad red PTs that relate to the hematopoietic cytopenia affecting more than one blood cell type. If I open erythropenia, I'll see the red blood cell disorders, red blood cell test results, hemoglobin decreased, hematocrit decreased. So that's where we're seeing erythropenia. And on the right-hand side, so I'm going to show you now using the browser in general. If I find an SMQ, I can put my cursor on it and I can open details and occurrences. On the right-hand side of the screen, it calls up information about the SMQ. We see that every SMQ has a numeric value, a number behind it. Here we see this is SMQ number 27. It always begins with a number 2. The name of the SMQ always has SMQ in brackets after it. That's to distinguish from a PT, which might have a similar name. SMQ in brackets means it's the SMQ. There's the level 1. The status is active and it doesn't have an algorithm. The SMQ description is a short summary of the notes that were taken when the SMQ was developed. So this is some of the information you'll see in the introductory guide, but you'll have more information in the introductory guide, the source that was used, and notes about testing, or if there's an algorithm that will be included in the notes. So this is information. Every SMQ has this. A right-hand mouse click on the name of the SMQ. Let me get it right. There we go. Open details and occurrences. You asked me, was it possible to export the SMQ? And if you have a look at the bottom of the box, you see that's the last option, SMQ export there. So if I click that, it will put the SMQ, hematopoietic cytopenias. It's put it in Excel. It's in my downloads immediately. It's open. There it is. It's in. And here is the SMQ. Let me just enable it for editing. I can expand the columns. There's the name of the preferred term. It tells me if it's broad or narrow. It tells me, is it the whole SMQ is active? It tells me a bit about when was that term added and has it been modified. A term might be added to an SMQ, it might be added and it's broad, and then as we get feedback, someone might say they think it should be narrow and it can be changed to narrow, this kind of thing. So you see there, this gives you an indication of the history of the SMQ. And then you see there the SMQ, it lists all the sub SMQs one under the other. When you do the export, the PTs are exported according to their sub SMQ there, the hierarchy. So that's, let me go back to the browser. That's export, whoever it was, somebody asked me about export. So this is the hierarchy. Hepatic disorders is an interesting one. It is a complex one, and I didn't go to it in the intro guide, but you can find out a lot more information in the intro guide. It breaks down into a level 2 SMQ, but then if I click one of those, for example, drug-related hepatic disorders, it breaks down even further into a level 3 SMQs. So even within that category or that grouping, it breaks down even further. Drug-related hepatic disorders, severe events, that one is, if I look at the details of that, that's a level 3 SMQ, and that breaks down even further into a level 4 SMQ before I then see the preferred terms. So it's not meant to confuse you. It's there to help you to be more specific in your search. Because hepatic disorders is such a big topic, we've subdivided it to help you. And when you run the SMQ, you can just look for hopefully the conditions that you're actually interested in, and not any hepatic disorder. So that's hierarchy. Let's go back now to algorithm. Remember the algorithm that I showed you with the anaphylaxis. So anaphylactic reaction is up here. This is the type of SMQ that's got categories. Remember, the broad PTs are put into categories, and the algorithm says, let's retrieve a patient who has something coded to one of these narrow terms, the name of the condition. And also, let's retrieve patients who have 2 or more symptoms that fall into 2 body systems. So here are all the respiratory symptoms. See, cough. I don't want to retrieve every patient who has a cough. You can't think that every patient with a cough has anaphylaxis, but if they have a cough and they come down, if we look down, they have category C, the skin, they have eye swelling or eyelid edema, then we start thinking, or maybe they do have anaphylaxis. That's why the algorithm looks for patients with 2 symptoms from 2 body sites or more symptoms: cardiac arrest, cardiac insufficiency. And you see here the category is indicated with the little letter D at where you see the PT icon. So that's anaphylactic reaction for an algorithm. Another algorithm, a good one to look at as understanding the logic, is acute pancreatitis. Acute pancreatitis has the narrow terms. Then it has category B. Let's look at them. What do these all have in common? They're all lab results, aren't they? And then category C are all symptoms. So what's the algorithm going on here with this one? We have different algorithms for different SMQs. So right-hand click, open details, and we'll see at the bottom, or you could look in the introductory guide in the document, SMQ notes. A report is considered a relevant case if it includes a term from category A, which are the narrow terms, or at least one term from category B, which are the lab values, so a patient with a lab abnormality, and also a sign or symptom of acute pancreatitis. So that shows you how the algorithm there is applied. Good. So we've spent quite a bit of time on that because this is, I think, is the most important concepts of the session today. But I'm now going to show you how you can use the SMQs We've talked about the inactive PTs. The SMQ itself could be made inactive, and there was once one SMQ which was replaced, and it was adverse pregnancy outcomes, and it was made, the whole SMQ was made inactive, and it was archived. It was then replaced with another SMQ, which was pregnancy and neonatal, relating to pregnancy and neonatal. So the inactive is similar to currency, but the terms are still current in MedDRA, but they can be switched off within the SMQ. That's what that one says. Versioning. Remember that it's important you use the SMQ of the version of MedDRA that matches the version of MedDRA that you used for coding the data. Use the SMQs with data coded with the same version of MedDRA. Because otherwise, if you use an SMQ that's from this current version of MedDRA, a new preferred term recently added to MedDRA may have been added to that SMQ. And if you use it to run on some data that were coded maybe 3 years ago or with an older version of MedDRA, you're going to be looking for cases that haven't been coded because the preferred term wasn't available. So you're going to have a mismatch. If you're using an old SMQ and you've got cases coded now, you might not have the preferred term in your SMQ and you miss cases. So be aware of this. There's an example. The PT, hormone receptor positive breast cancer, was added in version 23 to breast malignant tumors. If I was using an old SMQ, I wouldn't have that PT in my SMQ, so I would fail to identify cases coded using a database coded using the later version 23. Here's showing you the benefits of SMQs. So as we've seen, they can be applied across multiple therapeutic areas. Doesn't matter what your drug is. Your anaphylaxis is anaphylaxis, whatever type of drug it is. So it's validated as well. It's been tested in at least one data. We say that the SMQs are tested on at least one regulated database and one industry database. So they're well tested. The exception to that was COVID-19 because there weren't data available for testing at the time. So apart from that one, the others have all been validated. They're reusable. It gives standardized communication. We've talked about that. Consistency. And a big benefit is that we do the work for you, or our experts and our teams out there in the industry, who are on the committees and the working groups or propose the SMQs. They've done the work for you. They're maintained and they're built centrally, and we maintain them every 6 months by reviewing whether PTs need to be added to them or not. However, there are 110, but these don't cover all medical topics or safety issues. And that's why in last week's session, I taught you how to build a query yourself if there isn't an SMQ available. And also, we take feedback from our users relating to the SMQs, and the PTs can be, new PTs can be added, or PTs can be made inactive once they're out in production. So here's a poll for you again. Which measures will increase the specificity of the SMQ output? So we've talked about a few of the characteristics. Which of them will make the SMQ output more specific? So only retrieving relevant patients, we hope. Which of these? Is it making use of the hierarchy of the SMQs, using only the narrow PTs, making use of an algorithm, or removing PTs if they reveal too many irrelevant cases?
Jane, can you just remind us, when do we use the research bin and when should we export the SMQ?
You export the SMQ if you want to use the SMQ and you want it in an Excel format. I'll also show you how you can take it from the version release. the research bin is used for building your own query. That's what we did last week. So the research bin is used for building a query. When you export the SMQ, you export it directly. You don't need to put it in the research bin for export.
Thanks very much. And are you going to show how we apply the algorithm while selecting a list of terms?
Yes, in the browser, I will.
Thank you.
So let's look at these answers. Does making use of the hierarchy make your output more specific? Yes, absolutely. Remember the hepatic disorders. I showed you using a sub SMQ will retrieve only some types of hepatic disorders. Using the hierarchy with the leukopenia, erythropenia, it makes it more specific. Using the narrow PTs, yes, again, that one definitely does make your search more specific, because you're only then retrieving patients who have an event which is highly likely to be the condition of interest. Making use of an algorithm, again, think about what I was saying to you about combining symptoms. Combining symptoms gives a doctor a more accurate diagnosis. Combining symptoms makes our search more accurate, retrieving relevant patients. Remember what I showed you about the PTs in the anaphylactic reaction. Not every patient with a cough has anaphylactic reaction, but maybe if they have a cough and they have angioedema, they may have anaphylaxis. And removing PTs. Now, this is an interesting one. I'm glad this is less commonly popular. We shouldn't remove PTs from an SMQ just because they're retrieving irrelevant cases. If once we remove PTs, then that makes the SMQ non-standard. We can't say that we are then using the SMQ as a standard if we've removed PTs. We may choose not to include those patients in our submission, but we don't take the PT out of the SMQ. And that's what comes into customizing searches that I'll show you later at the end. Stay with us and I'll show you at the end. So I would say of these, the first three and the last one, I would say is not really going to increase the specificity. It's the review of your patients, which is that's next stage rather than the SMQ output itself. Browser view. Well, we have shown you the SMQs. So let's see where we go next. So using the SMQ now within the MedDRA browser. I described all the types of browsers we have. I told you all that ahead of time. This is the slide that describes it. We also have a mobile browser. It's not an app, it's a URL, but it's structured with a bigger interface that makes it clearer for you on a screen of a phone or a tablet. So this, I won't go through this again. I've told you all about the browsers, and here are the links you need to access them. All the browsers require your MedDRA ID and password. We know that we can search MedDRA and the SMQs. We can use all the MedDRA languages, see the interface in language. We can use the research bin for building a query. And if you want more information on that, come along to our other webinar. The browsers show both primary and secondary links. They allow us to upload terms that we run against the SMQs, and that's where I'm going now. They allow us to search with an advanced search, and the web-based browser allows you to see the supplemental terms coming in the next release. But I would say there's a little caveat. If you are interested in supplemental terms, look at them in the legacy web-based browser, because that's where it works best. It doesn't work quite as we'd like it to work in the new web-based browser. So if you're interested in supplemental terms, go to the legacy browser because you'll find it easier. How to run SMQs. Well, think back now to that diagram. You remember that diagram of the database on the left-hand side and the clipboard and then the list on the right-hand side. So from an IT perspective, an SMQ is a stored query. It's a list of preferred terms. That's all it is, as I showed you on the diagram. Most organizations would store the data as LLTs. That's what's used for coding. But the ASCII files for the SMQs include both the PTs and the LLTs together. So you load your SMQ into a query tool. You might have a commercial database, but if you don't, I'm going to show you how you can use the browser to do this. And you're looking for hits. Remember the diagram with the clipboard? You're looking for a match between the PT in the SMQ and the PT that was used for coding of that patient, or the LLT that you actually coded to, and the LLT that sits below the PT in the SMQ. It's optional. You can use the distinction between narrow and broad. You can apply an algorithm or not. You can just use it as a broad list, and you can use the hierarchy, purely options for you. So now let's think of an actual practical question. If I have a very small database here with 17 patients, and they've got events, we can see them listed. They've got multiple events each. We can see their case ID. How can I run the SMQs to look for potential cases of interest? Now I'm going to go to the browser and use a feature called the SMQ analysis tool. This feature is available in both the legacy browser and the new browser, both the desktop and the web browser. And it gives me the option to apply any of the characteristics I've described. So let's do this. Let's do it in practice. So let me just bear with me while I just close some of these things. Let's get this sorted so I can see. So let's have a look. First of all, let's go to the legacy browser. Okay, the legacy browser. So that's why the screen is now going to look slightly different. I'm going to open on the right-hand side web-based browser. It's also available as a quick link at the top. You see what I mean now about the screen being more cluttered, less clear. So I switched to I can switch to SMQs just the same as I was before in the other browser if you want to see the SMQs. But what we're interested now is a function called SMQ analysis. And in this browser, it's a blue button, top of the screen there. SMQ analysis is a blue button, and we get a screen like this. Okay, with that. And now I'm going to show you again to do the same thing in the other browser, the new web-based browser. This is the new web-based browser. So in order to run, I'm using this one because it's clearer. Top bar, navigation bar, look at the icon of the screwdriver and the spanner or wrench, you might call it. When I hover, it tells me that's tools. Click on that, and I have 4 tabs. I have the MVAT report, MedDRA version analysis tool, MVAT data impact. We talked about this earlier today. This is where you compare any 2 versions of MedDRA and get a report showing the differences between the 2 versions of MedDRA. Then I have an option for hierarchy analysis. I showed you this last week. So that's something if you want a refresher, watch back. And SMQ analysis is what I'm going to show you now. So I click on that tab and I see here all the SMQs listed. So if I don't choose an individual SMQ, what will happen is I'm going to import my data and I'm going to run all of the SMQs against my data. This is a screening exercise, maybe to see if there's a pattern. Is there a trend? Do my patients have any of these conditions? That might be something you do early on in your development of a drug or clinical trial. So let's change it back to version 28 of MedDRA so we match what we're doing today. I'm going to run, you see the drop down, I can run narrow, broad, and algorithmic. I'm going to run broad. And just to show you, I'm going to leave all of these defaulted so that I'm going to run all of them. Now I'm going to open a spreadsheet showing you the data. Let me show you on the desktop. Actually, I've got it on. No, this wasn't what I wanted to be. So here I've got a spreadsheet here, SMQ analysis exercise. This is what my data looks like in the spreadsheet. This is the standard format that you need, and the tool will tell you about the format. This is what I have. You saw it on a slide. I'm going to import this spreadsheet into the browser. Okay, so let's go back to our browser. Here we are, the SMQ analysis. So I'm going to navigate now. There's the format guide telling you what the format is. You don't need the code and the term. You only need one or other. They're optional. Also, row ID may help you match it up against your patients. So now I'm going to look for that spreadsheet. It was on my desktop, so let's find it on my desktop. SMQ analysis exercise, there it is. Navigate to that. And immediately I've got the results in my download. That shows just how quick it is. So here we see the events have been imported and they've been matched against every single SMQ to see, is there a pattern? So the best, I could scroll down and look at which SMQs have been matched to which events. And it tells me whether the SMQs that have been matched are hierarchical and whether they're algorithmic or not, output there, or hierarchical. But the best way, what I would say, is to make it meaningful is to apply a filter. So this is basic Excel, sort and filter. I'm going to apply a filter to the row, the row with the headings, and then I can filter here and select one SMQ in turn. So let's have a look at acute pancreatitis, and then we can look at this. I haven't applied the algorithm here. I've just applied it as a broad SMQ. So I have 4 events matching the preferred terms in the SMQ of acute pancreatitis. Let's scroll across. 3 of them are broad preferred terms and 1 of them is narrow. Patient 1 had amylase abnormal and abdominal distension. So they had an abnormal lab result and a sign or symptom, there of abdominal distension. Patient number 7 only had abdominal pain. They only had 1 symptom. And patient number 9 had a diagnosis of pancreatitis acute. So if you can think back to what I showed you with this algorithm, and then we'll repeat it again and you'll see what I mean. So we've retrieved, by running just the broad search, I've retrieved 3 patients, 1 with a lab result and a symptom, 1 with a single symptom, and 1 with a diagnosis. And you could do that. You could filter, you show your results. We could be here all morning looking at the different results of the SMQ against every SMQ. So let's go back and do it again. So I'm going to go back to choose my SMQ. Now let's just run acute pancreatitis and let's run it as an algorithmic search. You see, I've changed the drop down to algorithmic, and I focused in on just now running acute pancreatitis. I'll do the same again. I'll find my SMQ analysis exercise again and open that. Exactly the same as I just did, but I've now just focused in on 1 SMQ. So this is the result now. So what's the difference? By applying the algorithm, I'm only seeing patient 1 and I'm seeing patient 9. So patient 1 had an abnormal lab result and a symptom. So they are relevant because of the algorithm. Remember the algorithm, and it reminds me there's a misspelling there in that column, but ignore that, please. You see there the hierarchy, the algorithm is listed, and then patient 9 had the diagnosis, so we definitely want them. But we've lost patient 7 who just had abdominal pain. That's because abdominal pain could be anything. So by applying the algorithm, we haven't retrieved that patient because they didn't have an abnormal lab result to go with it. So I've explained that in quite a lot of detail. So I hope it's clear for you. If you've got questions, let Leander know and we'll stop. So this is, you have it on the slides showing you what I've just done. I've chosen it in the new browser, but you've got both on the screen. There's the input, there's the results. And that's what happens when I applied the algorithm. We looked there, that one's doing, I don't know, several SMQs together. I just focused on the pancreatitis, and it shows you there the algorithm. So you've got all those slides as a reminder to take away. Any questions, Leander? I see we've got half an hour left.
Yes, please, Jane. Could you just show again the information that you need to run the report? I know it shows it in the screen in the new web-based browser, but could you just show again the information that's needed? Thank you.
What, you want me to run it again, do you? Is that what you want me to show you how to run it?
It's the template, that's it. The
Oh,
screen you're
yeah.
showing now. So you can see the row ID, the LLT, PT term.
Yeah. Let me tell you. So the whole process was go to the tools on the navigation bar. It's defaulted in, so you'll see it. Or if you're in the legacy browser, it's a blue button at the top right-hand side, SMQ analysis. Here within the new browser, you've got 4 tabs because you've got 4 analysis tools together. I want the last one on the right-hand side, SMQ analysis. I click on that. SMQ analysis. I click to firstly choose what version of MedDRA, what type of search. Am I doing broad, narrow, algorithmic? And I can select all the SMQs by default or select one. Click next. It takes me to show the format and I can navigate to where I've stored my data. And then I choose the file and it does it immediately. Okay, hopefully that's clear for you.
Thanks, Jane. And what does the row ID mean in your example?
Row ID is just an identifier for yourself. That just is, maybe you want, it can be a number, it can be anything. This is just to help you match it back to your data, your Excel. You might have a database, you might have Excel, and because a patient can have multiple events, it's a row ID. It's just an identifier to help you combine it with your database. It's optional.
Thanks, Jane. And just one on personal data. So when you imported the data, what happened to it?
It doesn't go anywhere. We don't want your data. We don't take it. We're very aware of that. You're just using the tool on your own system. You're using the tool on the web. It doesn't go anywhere. You're just applying our analysis, our code, if you like, to your data. It doesn't, we don't receive it. We can't see your data. So I mentioned at the beginning when you asked me about, was it only clinical trials? Well, the SMQs can be used for clinical trials, maybe early on as screening, I already mentioned, or it might be that you have an idea, preclinical results or a class effect, and you know that you have a specific SMQ or SMQs you want to run on any of the clinical trial phases. Post-marketing, you can use selected SMQs to retrieve cases for a suspected or known safety issue. You can use them for signal detection, again, a kind of screening again by maybe running all the SMQs. You can also program them and use them for single case alerts. So if a patient is a case is received where it matches a condition that you're interested in, it can give you a flag, an early warning that a patient's been received, and you can then retrieve that review that patient early on when they're first received, or you might want to give them a questionnaire, a guided questionnaire for whether it's the severe cutaneous reactions, or you can follow up with that patient more quickly. You can use them for periodic reporting, aggregating cases for safety, and lack of efficacy is one which is commonly customized, and we're going to come back to that. So now let's look at two fictional data analysis scenarios. Scenario 1, your company has started development of a new drug for onychomycosis, projected to take 8 years and cost $1.2 billion. What is onychomycosis? Well, it's a toenail fungus. So it's not really a life and death severe event, but it's very can be very uncomfortable, nasty for the sufferer. So in this scenario, as part of safety surveillance, you decide to periodically run the SMQs for serious adverse events against your data. And you identify 5 possible cases of hemolytic anemia in your 1,000 patient exposures. Now, hemolytic anemia is a severe condition coming from something which is used for not a particularly bad condition. So this gives us an unfavorable risk-benefit ratio because the drug is for a nail fungus. So your suggestion, based on your results, is to stop development of this very expensive project. Your management reviews follows your recommendation. It saves time, money, potential patient risk. Job well done. You get a pat on the back, you get a bonus for that in recognizing it so that you could stop the drug development early on. Very courageous move, but it was evidence-based, based on the SMQ results. Scenario 2, you're developing the 4th drug in a therapeutic class. So this is a Me Too drug, but other products in the class are associated with development of high blood pressure and angioedema. But now it's not for toenail fungus, it's for a more serious target disease, whatever that may be. And so based on that, this is an acceptable risk. During development, you run the SMQ of hypertension and angioedema against the data early on in the post-marketing of the product and development. Over time, it becomes clear that your drug is not associated with either of these risks, like the other drugs in the therapeutic class. This gives patients a safer alternative, so it gives a relative benefit. And this is what I was saying about regulatory authorities being able to compare products for the same therapeutic effect from different companies. Congratulations, well done again. You get another pat on the back and an even bigger bonus. So well done. So that's the section all about the SMQs, using the standard SMQs, concluding there with how they can be used. Now, before we part today, I want to show you customized searches. And I did mention, remember the poll? I said, don't take out preferred terms from an SMQ. Do not modify an SMQ unless you have compelling reason to do it because it makes it non-standard. You can adjudicate and review your patients before you submit them to retrieve any irrelevant cases, but don't customize the SMQ itself. If you do customize an SMQ because you have good reason, you must call it a modified MedDRA query based on an SMQ. That's the wording you use when you send it into an authority. Document the modifications and your rationale for doing so. Remember though, it's non-standard now, so you need to review it with every 6 months and maintain it because it's not an off-the-shelf SMQ. But just a note there that the SMQ that is commonly customized is lack of efficacy, lack of effect. Because if I have a drug, how do I know it's not working? What will the patients be like if the drug isn't working? What will they report? Well, depending on what the drug is for, they're going to have different symptoms, aren't they? They're going to complain of different things with the drug not working on my drug compared to your drug because they're for different target diseases. So to build a customized query, you might decide that there isn't an SMQ and you need to build a query from scratch. To do this, you need to have medical knowledge. You need to know your data, know MedDRA, know where the terms are in MedDRA. And for tips on this, if you weren't with us last week, You can find advice in the MedDRA data retrieval and presentation points to consider document. And when you have built the query, you can, I would say, save it for future use, but remember you need to maintain it so that the new terms being added to MedDRA or the hierarchy changes in MedDRA get applied to your query. And then think about sending it to us. Maybe we can work together on it and make it into an SMQ. And that was what has happened with many of our SMQs. So before we wrap up our summary then, let's go to the browser. No, let's go to the website first of all. Let me show you again how to use tools. We have a whole page for SMQs. Now on this SMQ page, we have, I have to do it again because it wasn't as quick as me in my clicks. Here we go, SMQs. Let's go to the SMQ page, standardized MedDRA queries. This will refresh your memory on what we've talked about today. There's the lactic acidosis SMQ, narrow and broad. On the right-hand side, we have a few documents. We have one called SMQ status. This lists all the SMQs as of September 2025. Let me just go back to where I was. There we go. Then the next one is an SMQ matrix. This one is an interesting document. This one gives you the whole history of all the work that's been done on SMQs, showing which ones of when they were released, when they were developed, when they were released. It also includes requests that we've had. Maybe an SMQ started and then it was discontinued. It may be at testing. There's one there, opportunistic infections. I had a friend who was on that team, actually, that one. It went through testing and it was discontinued. Others might have been requested and not taken on. So here the ideas for SMQs that have been rejected. So if you're thinking of submitting one, you can have a look here. You can see when it was rejected. Often it will show you when. I think it tells you somewhere when there. That one tells you when it was rejected and why it was rejected. So that's a bit of background for you. We won't spend too long on that because I'll get sucked into details and use up the time. Here we see a link to an example of an SMQ, case studies, the training materials, the support documentation. That was by, I went to case studies. No, I went to basics to get that. Case studies tells us case studies, training. There's also a link telling you here, it's just an advert for a book, a CEOMS book. The SMQs were a CEOMS initiative back at the start of the SMQ project. CEOMS and the MSSO on behalf of ICH were both looking for some way to group terms horizontally. If you imagine we've got the MedDRA hierarchy, they were looking for ways to group related signs, symptoms together with diagnoses and all the other terms that could relate to a condition. So CEOMS and ICH then worked together, and that's where there's a CEOMS publication. We call it the Red Book. It's basically the notes that were made when all the SMQs were developed, and I'll show you where you can download that information rather than buy the book. And also there's a document newly there that it just summarizes some discontinued efforts for a particular neurodevelopmental disorders, neurodevelopmental delay, SMQ efforts there. So that's the SMQ page. One more thing that I wanted to show you was, if I go back to my desktop, I put here somewhere let me go to the website let me logically, otherwise I'll confuse you. Sorry, bear with me. On the website, so if I go to the home page again, you have top right hand corner, you have a quick link called downloads. This is where you need to log on with your MedDRA ID and password. If you don't have those, you can get them from our self-service application, or you can ask your primary point of contact. And you can go to the downloads page. This is where on the right-hand side, you have the SMQ original documentation. That's the content of the book I was telling you, giving much more detail about each of the SMQs. And also here, this is where you download the MedDRA release. If I click on 28.1, we see all the languages. If I scroll down to English, I downloaded it just by clicking on it. It's now in my downloads. And this is what's in it. It's a folder, and this is what's in it. So these are the ASCII files that contain the MedDRA content, and this is what gets imported into your database to form the MedDRA database itself. This is what's in our browser. You use those for updating the desktop browser every 6 months, and you get them from the download section of the website. This is where you see the SMQ introductory guide. I mentioned you can get it here, or you can get it on our website. This is where you can see a version report of MedDRA listing the changes between the last version of MedDRA and this version. And the one I wanted to highlight for you is the SMQ spreadsheet. So when I click it, I need a password. This is an unzip password. I need this to open the folder. So this is not your standard password for the opening the browser. This relates to the actual version of MedDRA. And if you don't have it, ask your point of contact in your organization to get it for you. So I've got it open already to make it quicker for us today. Here it is. No, that's not the one. It was the other one. There it is. Sorry. Let's try again. This one's already open. I opened it before the session. So this is the spreadsheet I mentioned. So let's go to the first tab. The first tab lists all of the SMQs, 110 SMQs, and we can use the Excel, I don't know what you call these, openers, I think I'd call them. And you can see those that are hierarchical, because we're seeing the sub SMQs underneath the main level 1 SMQ. So this is just like, what would you call this, a map, an outline, a framework with the names of the SMQ, not the actual content. I need to go to the next tab, and the next tab, labeled SMQ 28.1, lists all of the SMQs. And again, with these Excel opener things on the left. And then when I open, I can see the actual content. One of you asked me, could I export the SMQs? You can. This is another way of accessing them in Excel. And then along the bottom, you've got summaries describing them, telling you which are algorithmic, which are hierarchical. You can see that. You can see the active and inactive terms. You can see the codes. So if you want to use an SMQ and you want to give it to maybe a statistical programmer or a colleague who does your analysis in the query tool for you, you can give these codes from here, or you can give them from the actual SMQ itself. So that's, I wanted to show you there from the website. So that, remember, that comes from downloads and then the actual release package that you're interested in. And that's in the file that gets downloaded. What else was I going to show you? Any questions, Leandra? Anything I've not shown our group that they'd like me to show them today?
Not show, but I did have a question. So your import file that you used, there are 4 columns required, but for one of our attendees, they were asking, can they have more columns of data? For example, further columns after D?
I would say no. I would say no. You can test it. The best way is to test it. I think the tool won't like more, but I've never tried it. But it's important that you have the headings. So even if you don't have those exact, you don't want to populate every column, use the headings. That's my experience of running the tools, is they want to see the headings. The tools like to see. I'd say probably no, but test it and try for yourself. I think you're better to keep your own data source and use the ID column so you can match it back.
Thanks, Jane. And on some of your information that you were showing, when you export the data, there's also a column called weight. What does that mean?
Oh, yes. Should we look at the weight? Yes, happy to do that. We've got a few minutes. So anyone who feels there hasn't been time or there wasn't time last week for questions, now's your chance in these last 10 minutes. Let's close this. I didn't show you the weight. One of our algorithmic SMQs, only one of the SMQs has weights. So bear with me and I'll show you. Systemic lupus erythematosus, SLE. This is kind of a nebulous, of a kind of a syndrome, if you like. And because diagnosis can be so hard and retrieving patients can be so hard, the way it's diagnosed is by counting up patients' symptoms and giving them a score. And that's what's used in practice. So that's what's been replicated in the SMQ, in the algorithm. So what happens is the algorithm scores the symptoms. So we've got our narrow terms again. These are highly likely to be the condition of interest. Look, different elements of the syndrome itself. Underneath, we have all these different symptoms grouped in categories, and it goes right down, F, G, H, many categories here. So rather than just combining the terms in the categories, the SMQ gives the symptoms in the categories a weight, a score. And then it adds it up. And if the patient has a score greater than 6 for their symptoms, that would constitute a case that you want to retrieve and review. So that's the way the algorithm works. It only retrieves patients who've got symptoms totaling more than 6. And that's what the weighting means. It means the categories all have a weighting and it multiplies up. It tells you here, you can read it from the browser, you can read it in the introductory guide to this SMQ, and it will explain it. I met somebody once who did a PhD on just this SMQ. So it shows there's a lot there. If you're working in this area, if it's an area of interest to you, you can really drill down and learn a lot from it and use the SMQ to serve your purpose. So let's see if I've got that spreadsheet still open. This one's got it. Let's go to this one. I'm going to go. So this is probably the one I think you meant. So this was the output when I ran my data against all of the SMQs together. It tells us which SMQs matched my patient events. And if there was a patient, I don't know if I have any in my data, here we are. These patients, well, let's filter. I'll tell you the easiest way to do it is to filter for that SMQ. SLE, systemic lupus erythematosus. So we see here that patient number 13 had a narrow, they had a term, SLE arthritis, which is a narrow term, so that doesn't have a weighting. It's only the broad PTs, the symptoms and things, which have the weightings. So patient number 14 had 3 events. They had a symptom from category H, F, and I, each of those 3. They had hemolysis, autoimmune nephritis, and double-stranded DNA antibody positive. 3 reports, 3 events, which are coded to separate PTs in separate categories. And based on their weighting, they had a score of 3 for the first one, 1 for the second one, 3 for the third one. So they then did have a score of 7, We know that that's greater than the cutoff threshold of 6. Therefore, that case was retrieved when that would be retrieved if we ran the algorithmic SMQ. Patient 15 had a score of 1 for photodermatosis and 3 for pleural effusion or 2 for epilepsy. So they had a score of 6. So when I first ran this spreadsheet, I didn't apply the algorithms. Remember, I ran a broad search. So if I was to go back, rerun this SMQ alone and apply the algorithm, I would expect to see patient 13 with their diagnosis, their SLE arthritis, narrow term. I'd also expect to see patient number 14 because they had a score of 7, but because 6 is the threshold, I wouldn't expect to see patient 15 because they're not above 6. I've explained that in quite a bit of detail, so I hope I haven't lost any of you. Any other questions, Leander?
Thanks, Jane. The indication is to select the PTs of interest in the beginning of a trial and run an SMQ analysis, or wait for the beginning of the trial and run the analysis against already reported events. So is it best to do it at the beginning, or is it best to do it at the end? Are we able to help with that?
We have to have data to run it on, so you can run it at intervals throughout the trial. If you're running a clinical trial and you have milestones and you might have a data safety monitoring board, or so you could run SMQs throughout a trial at agreed intervals, but you wouldn't start at I wouldn't do it at the beginning. I'd wait until there's at least some data because you've got to run it against data, and then definitely you'd run it at the end. But you could wait until the end to find out exactly what's happening, or you could run it at intervals if your trial is set up like that you have milestone or interim analyses, I think it's called, isn't it?
Thanks, Jane. And we had one from the hemopoietic leukopenia question from earlier, but I needed to clarify a couple of bits, but I've had that now, so I wonder if you could help with this question. If only the hemopoietic leukopenia was expected in my summary of product characteristics, and my PTs of the ICSR is hemopoietic cytopenia, can the PT be considered as expected or not?
I'm sorry, I can't. Pharmacovigilance processes is not really it's not my area of expertise, and it's not something that we would regularly advise on. I can take your question away and have a look at it, and if it is something that on reflection me or one of my colleagues can help you, we can get in touch with you, but generally we don't advise on expectedness or regarding that, relating to it. So you're saying if we run the SMQ of leukopenia and you have a report of hematopoietic cytopenia, is that considered expected? I don't, to be honest, I don't want to get into something today that I'm not confident on, because I've never worked in that area. Sorry.
Thanks,
I don't
Jane.
want to give you false information, but we will take your question away and review it offline.
Thank you. And could you just go to the website and show where to access the self-service application?
Oh, yes.
In order to retrieve your certificate.
I'll do that. I've got one. Yep, I've got 1 minute. So this is what we've done today. I won't read it out. You know what we've done because you've been with me on the journey. And at the end of the slides that Leander sent you, you've got some links, useful links to the website, the browsers, the change request tool, support documentation, and we've done your questions and answers today. And let's go to the website, and I'll show you where you can get your certificate. This is also where you get your credentials for accessing the browser, or the unzip password, or finding out who your point of contact is. The SSA is a self-service application. You can see along the top, if I look, I'm waving my mouse along. So credentials tells you your password. If you're not a nominated point of contact, you can't get the unzip password or the change request password. You can only get your browser password. So, but it will, the tool can tell you who your point of contact is. If you go into it with an email address from the official domain at your organization, and if our tool recognizes it, it matches our records for your organization, you can then see who your POs, who your point of contact is, and you can also get the basic password for the website and the browser. So that's there. The subscription status is for confirming which organization has a subscription. The retrieval point of contact is from their subscription contact. Training is where you go for your certificates. You enter the email that you used for registering for the class. It will send you a code in your email just to validate your identity. That's so nobody can steal your certificate. And then when you enter that code that comes by email, you then will retrieve your training history record, your training record, and your certificates can be downloaded from there. And the certificates are there for 3 years. So you can look back at other webinars you may have attended in the past. Just remember, if you change organizations, you have to let us know so we can update your email address on our system. And that's everything. But give us 48 hours because the system needs 48 hours to refresh and process your password. Whew, I make it 11:02 here in the UK, so I'm keeping people now. Any final thoughts or comments or questions, Leander, before we let everyone get on with their day?
I've been scanning the questions, and I think that's everything. We've had people now saying thanks for the presentation. So I want to thank everybody for all your questions. I hope we managed to answer them. There's one we're going to take away just to have a look at it afterwards. Yep. But it was great to have an interactive audience. So thank you very much.
And we're here the whole time. So if you've got questions, just email us up to the help desk, any questions, and we'll follow up with you, any questions, if something occurs to you when you get offline. So thank you all. Thank you for your help, Leander. Thank you all for your attention. Enjoy the rest of your day and stay safe, everyone. Bye-bye.
Bye-bye.